To cut axons using in vivo two-photon imaging, position anesthetized zebrafish embryos within a drop of agarose as it hardens in a specialized chamber consisting of a Teflon ring and cover slip. A glass slide is placed on top of the chamber, which is then flipped upside down. Prior to the axotomy, take a before image of the axon you wish to cut at 40x. Zoom into 70x on the region you wish to axotomize, and with the laser off, turn up the laser power. Briefly, turn on the laser. In the after image of the axon, you will see scattered axonal debris if the axotomy is successful.
Hi, I'm Georgianne O'Brien.
Hi, I'm Sandra Rigo.
And I'm Shauna Martin.
And we're from the laboratory of Alvaro Sagasti in the Department of Molecular, Cell, and Developmental Biology at the University of California, Los Angeles. Today we'll show you a procedure for two-photon axotomy and time-lapse confocal imaging in live zebrafish embryos.
We use this procedure in our laboratories to study degeneration and regeneration of peripheral somatosensory axons.
So let's get started.
To begin, we'll prepare a 1% low-melt agarose solution for embedding. We dissolve the agarose in double distilled water by heating in a microwave before aliquoting into small tubes and storing in a heating block at 42 degrees Celsius. First, embryos can be placed into a 5% PTU ringer solution at 22 to 24 hours post-fertilization to inhibit the formation of pigment, which is brightly autofluorescent and will obscure axons on the two-photon microscope. Next, we select embryos for imaging and remove their chorions by gently pulling them apart with forceps. Once the embryos are selected, they are anesthetized by adding about 0.02% tricaine to the PTU ringers. Before proceeding, make sure animals are not responsive to touch. Prepare the long-term imaging chamber by applying vacuum grease to one side of a glass or Teflon ring and fixing it onto a glass cover slip. Taking care not to transfer much of the PTU ringers, transfer one embryo into the 42-degree agarose solution with a glass pipette. Then, transfer the embryo with one drop of agarose onto a prepared cover slip. Once transferred, position the embryo as desired while the agarose hardens. Keep in mind that the imaging chamber will be flipped when you're done so that the cover slip will be the top surface. If you have a motorized stage and can image multiple locations at once, repeat these steps for each embryo. Allow the agarose to completely solidify, then fill the ring with 0.02% tricaine PTU ringers. Finally, apply vacuum grease to the other side of the rings. Then cover them with a glass slide. Flip the seal chambers over. Now we're ready for two-photon axotomy imaging. To prepare for imaging, place the mounted embryos on a slide holder under the microscope. Focus on one embryo with a 40x 0.8 numerical aperture water objective, then turn on the laser. Set the laser to a wavelength of 910 nanometers and a power of 30 milliwatts. Open the imaging software. We use Scan Image software developed in Carl Swoboda's laboratory. At this power, one can scan the field for a candidate axon that will be severed. Once found, take an image of the target axon, mark the first and last Z positions, acquire the image, and make a maximum projection of the Z stacks. Next, zoom in 70x on the branch of the axon you wish to axotomize. Turn up the power to 180 milliwatts and set the number of Z slices to 1. Initiate a single scan with the laser by pressing Grab. This should be sufficient to sever the axon. However, it is necessary to optimize this procedure for your microscope and experimental goal. Finally, zoom out, reduce the power to 30 milliwatts, and take an image of our newly severed axon. If a custom-built two-photon scope is not available in your laboratory, the Zeiss 510 confocal two-photon microscope can also be used to sever axons. We begin by placing the mounted embryo onto the stage and bringing it into focus using a 25x water objective. Next, Turn on the two-photon and argon lasers in a multi-track setting so that it is possible to switch from one to the other. Although both lasers are used to detect GFP, the two-photon emission is visualized with red and the argon laser emission with green in order to differentiate the two. Use the argon laser to identify an axon to injure. Under Z settings, mark the first and last optical sections, take a confocal image, and create a maximum projection of the Z stack. Turn off the argon laser and turn on the two-photon laser. Scan at an intensity of about 9% transmission to make sure that the axon is still in focus. Click the stop button so that the crop tool will be available. Use crop to zoom in on the area of interest. Usually, we zoom to about 70x. Choose the region of the axon to be injured and bring this region into focus. The zoom can be checked under the Mode tab. Next, under the Channels tab, change the intensity of the two-photon from about 9% transmission to 15 to 30% transmission. To activate the new settings, click on the Fast XY button and then click Stop quickly afterward to avoid excess damage. The axon should be seen as scattered debris if the procedure worked. Finally, to ensure that the axon was indeed damaged, switch back to the 488 nanometer argon laser. Take another confocal image and create a maximum projection of the Z stacks. Now that we've shown you two ways to sever axons using two-photon laser microscopy, we'll look at confocal time-lapse imaging of regeneration using the Zeiss LSM 510. To prepare time-lapse imaging, be sure to turn on your heated stage at least 30 minutes before setting up your time-lapse movie. Place the mounted embryos on a slide holder under the microscope. Focus on one embryo with the desired air objective. We use a 20x 0.5 numerical aperture objective. Open the Zeiss LSM 510 imaging software and then turn on the appropriate lasers and set up the desired configuration. Open the Scan, Stage, and Multi-Time windows if you have multi-time software. Define the position and configuration of your first embryo in the multi-time window. Activate Fast Scan on the scan window, then move the stage to the desired XY position. Mark the first and last Z positions in the scan window, press Stop, and then Mid, also in the scan window. Wait for the scan of the middle Z slice to finish. Then press Mark Position in the stage window. If you are only imaging one embryo, click on the Single Location tab in the Multi-Time window. Click on Replace XYZ, then click on Save Configuration. Agree when asked to override the previous configuration. Now you can set up the parameters for image acquisition in the Multi-Loc window. If you have a mechanized stage, you can set up multiple locations to image multiple embryos. In this case, you should select the Multiple Locations tab before defining the XYZ and configuration for your first location. Also, be sure that the pull-down menu just below the Multiple Locations tab is on location 1 and that the pull-down menu in the configuration section says Multi-Loc 1 before you click on Save Configuration. Define the position and configuration of your remaining embryos in the multi-time window. Locate your next embryo, press Fast Scan, then move the stage into the desired XY position and mark your first and last Z positions in the scan window. Press Stop, then Mid, also in the scan window. Wait for the scan of the middle Z slice to finish, then press Mark Position in the stage window. Click on Replace XYZ. Check that the pull-down menu just below the Multiple Locations tab is on location 2 and that the pull-down menu in the configuration section says Multi-Loc 2. Then click on Save Configuration. Agree when asked to override the previous configuration. Repeat this process for the remaining embryos. Set up the parameters for image acquisition in the Multi-Loc window. Choose the GR/GL option in the List of Blocks section, then enter the desired number of group repetitions. This is the number of times the confocal will acquire an image at each location. Enter the desired wait interval in the Parameters section. This is the amount of time from the start of one imaging repetition to the start of the next. Select Z-Stack XY in the Configuration section. Enter your base file name in the bottom section. Click on Select Image DB and choose your database. Click on the Options button, then select your MDB folder to save temp files to. Check Keep Final Image Open, Save Final Image, middle of the Z-stack, and select Wait Interval. Click OK to close the Options window, then click on Start Time. Leave the multi-time window open for the duration of imaging. Now it's time to analyze your data. When the time-lapse imaging has finished, the multi-time software will compile all of your temporary files into a summary file for each location. If you want to stop the movie before it is complete, be sure to press Finish instead of Stop so that a summary file will be created. The temp files and summary files should be automatically saved to the designated MDB folder. Here is a representative example of a time-lapse confocal movie following axotomy at 54 hours post-fertilization. The first frame shows a dorsal view of the axon before axotomy with a yellow arrow pointing to the axotomy site. The following time-lapse movie begins within 1 hour after axotomy and lasts 12 hours. One confocal stack was collected every 20 minutes. This time-lapse confocal movie is recorded using the same parameters at 78 hours post-fertilization. The first frame shows a lateral view of the axon before axotomy with a yellow arrow pointing to the axotomy site.
We've just shown you how to generate precise tissue damage and visualize live zebrafish embryos using two-photon axotomy and time-lapse confocal imaging.
When doing this procedure, it is important to optimize the amount of power used for two-photon laser axotomy to avoid causing excess damage.
So that's it. Thanks for watching and good luck with your experiment.